What's up, ladies and gentlemen, and everybody in between? I got a tortilla here. Why would I have a tortilla? Half-eaten tortilla. When I'm talking about neurons, well, you're going to find out here in a minute. In the meantime, I'm going to eat this tortilla. Just a plain tortilla. In this one, we are going to talk about how do these neurons send these signals. So our nervous system, all of this it's made out of a lot more cells than just neurons. We talked about those. They're called glial cells, and we go way out here, and we go back to tissues. We got our neuron, our axon, our dendrites, and then we got all these other cells all around it called glial cells. Right? We got our astrocytes, which create our blood-brain barrier. We'll talk about those. We got our oligodendrocyte, which I'd say insulate the neuron. We're going to talk about those. There are actually cells down here as well. I'm going to talk about those, but they're actually this little tortilla. And then we got these ependymal cells, microglia, and then we also have the neurons, which are these things in blue. How the heck does that result in thinking? It's pretty crazy. You're telling me you can store memories and think thoughts based on cells? That's weird. That's really freaking weird. And it's going to get even weirder because I want this to be the kind of question you ponder here. How do neurons send signals? We're going to learn that. And most people will say it's this, it's electrical. Maybe a few would say this. And probably none would say this. But by the end of this, I hope that you see that this is actually, it's all three. It's all three. It's electrical, chemical, and mechanical. All right, so the signals come in here at the dendrites. They then must, if they go out here, they must travel the entire length of the cell. So let's talk about how a cell does this, how a neuron does this. And we're just going to kind of zoom in. And we're going to draw an old friend, actually. We've seen this guy before. We're going to draw an old friend. So we're doing a little zoom in of this. So this would be out, the outside of the cell. Outside. This would be inside. Sorry if you can hear my chewing, but I need a little snack. All right. And we're going to draw an old friend. One of our favorites, one of my favorites, this guy. This guy is a sodium potassium pump. Sodium potassium pump. NAK pump. Whoop, that's not what I asked for. Sodium potassium pump. And what does this guy do? I'm sorry. Put that like that. Thank you. That's what I want. Goes right there. This guy, take a decay. Take a decay. And let it decay inside. Oop, don't want that. Want this. Bah. pumps it in. And it pumps in 2 at a time, kk. And it results in a lot of K on the inside. A bunch of K. I'm just going to cut it back down to 1. And I'm going to just make it real large. So this results in lots of K on the inside. Lots of K on the inside. Potassium be blanging all up on the inside. Okay. Now, does the cell want sodium on the inside? Nah. Nah, bruh. It does not want the sodium in the inside. Nah, doesn't want it on the inside. Where's it got to send it then? Got to send it to the outside. This results in a lot of sodium on the outside. Lots more sodium outside compared to potassium. Why is that? Why is there more sodium outside compared to potassium? Well, because pumping out 3 for every 2. Right? 3 pluses. 3 plus 3 positive charges go outside. 3 positive charges go outside. 2 positive charges go inside. What just happened? Bitcoin stole my computer. 2 positive charges go inside. All right. Make it a sense, right? 3 positive charges go outside, 2 positive charges go inside. Keep my language here the same. All right, you extrapolate that out a bunch, right? That means this is going to be more positive compared to this. Okay, so the outside of the membrane is going to be positively charged. And the inside of the membrane is going to be negatively charged. Oh, that doesn't look quite right. There we go. In comparison, right, relatively speaking, all right, this pump, if you remember, it's a mechanical pump. So it actually opens and closes like this. Biomechanics. And I've got Drake here from Hotline Bling being the sodium potassium pump. And you see how he's moving. He's moving and shaking. And the way I like to think of this is he's a bouncer. And it's ladies' night at the club, right? This is how you can remember the orientation of which way we send things. You always want more ladies than you do frickin' bros at the club on ladies' night. So you bounce 3, nah, you can't come in. You bounce 3 and you let in 2 ladies. Okay. You come inside, okay? Okay. Nah. Too many frickin' Y chromosomes on the floor tonight. We gotta kick out 3 dudes. You'll never forget it if you remember that. Nah. Outside. KK, come on in. All right. The point being here is that it's a mechanical pump. You know, just like ATP synthase spun, this thing goes like this. So you can't send signals down a neuron without this first set up. And this is a mechanical process. It's called your sodium-potassium pump. And I don't know if you remember this, but this is in all of your cells. Helps keep the inside different from the outside, which is one of the definitions of being alive. And about 1/3, I think it's 1/3. It might actually be 2/3. My brain is not working. I want to make sure I give you guys the right number here. I'll be right back. I'll make sure I get this right. All right, I was right. About 1/3. 1/3 of all your ATP goes to pumping, to sodium potassium pumps. That's how important these things are. Sodium potassium pumps. And they're extremely important in your nervous system because they set up this membrane potential, or the chemistry of having a lot of sodium outside and potassium inside. This is critical because this is how we send signals. Okay, so now let's just think about that. Let's just think about the chemicals, the particles, the ions, the elements, the atoms. We can think of all that as the chemical nature. You can also think of it actually as mechanical or also electrical, but we're first going to just think of it as the particles, the chemical particles, the atoms. All right. So let's draw a couple of, and how does this neuron send these signals? That's what we're going to talk about here. All right, so we got a membrane there and a membrane there. All right. Well, what happens is sodium opens up a channel. Sorry. Let me talk about it this way. All right, we've got boom, 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 and boom, boom. This is going to be a sodium channel. Sodium channel. Okay. Nice and easy. All right. We will also have potassium channels. Draw those in a different color here. Let me draw them a little closer together just to be clear. So these are two proteins that will open up like this to allow ions to come through. All right. Beautiful. Digging it. All right, so this is a sodium channel, and this is a potassium. And you'll probably want to draw these big. We're going to be drawing a bunch of these. Like you could do this whole thing on one page, this kind of part. Not this, but we're going to be drawing like a series of these. Just to give you a little heads up. And a potassium channel. Okay? And normally, we got lots of K up in here, right? Lots of K up in here. Lots of K. And we got lots of sodium out here, Na+. All right, and when that's the case, these are all closed. But, and we'll talk about where this comes from, some of the sodium now is inside. It's come from the dendrites. It's come from another neuron sending it a signal. And we'll talk about how that happens, but we're just in the middle of that process right now. So what happens is this sodium comes over here, and when it gets close to this channel, it opens up. Boom. So now it's open, and this allows more sodium to rush in. Okay? Like so. All right? And when that happens, what do you think is going to happen? Sorry there. I'll make a little copy of that. What's going to happen? This sodium channel is now going to open. Okay? It's pretty nifty. Let me get rid of this and get rid of this. Okay, then what's going to happen is that sodium's now diffused. Sorry, we still have that. This is now open. And so more sodium is going to be able to rush in here. Right? Like that. All right. What's going to happen eventually over time, let's just keep this cruising on down, and you guys can pause me if you need to draw any of that out. I'm going to keep just cruising through this. Eventually, this will be full of sodium over here. So what will happen is that as this membrane potential kind of changes, and we'll talk about what that means, this is going to close, and this potassium channel is now going to open. And it's going to kind of chase the opening of the sodium channels. So it's going to chase it, and now it's going to leave. Like so. All right? And then the same thing is going to happen over here. So as sodium reaches this channel, it's now going to open. So sodium will now rush in here. Oops, not green, blue. Sodium will now rush in here. And open up the next channel. Meanwhile, this will begin to close. This will begin to open. This will now be closed. And I'll walk through this once we kind of finish drawing it out. And then this will open like so. Okay. And that process kind of just continues on. And I'll write down what happens here to kind of summarize this. So what happens is when sodium gets near a sodium channel, it opens it up. And then sodium can rush in and open up the next sodium channel. Okay? You can see that here. Sodium rushes in, opens up the next sodium channel, so on and so forth. After a while, this sodium channel will close, and the potassium channel will open. And then the potassium will leave. So we have sodium coming in, potassium leaving behind it. It's like a wave that propagates. Sodium opening, sodium coming in. Once that leaves, then it's potassium opening, potassium leaving. All right, and then it just propagates out. So let me put some words next to these. Okay, sodium channels open, more sodium channels. not sodium channels. Sodium opens, think of it that way. Sodium opens more sodium channels, allowing sodium to rush into the neuron. Okay. After some time, sodium channels close. We'll talk about kind of why that is. Sodium channels close. And potassium channels open, allowing potassium to rush out of the cell. Pretty cool. Pretty cool. Okay. Then this process continues on. You could really just say, and then we could just say like process continues. Process continues. This is called an action potential. This is called an action potential. Why is it called a potential? Because it has to do with electrical properties as well. So you can think of this as chemical or mechanical, because there's opening and closing of channels, and there's mechanical movement of chemicals, of atoms, right? It's chemistry. So you can think of this as both of those that's occurring. And it's a propagation. It's a wave. One wave of sodium followed by a wave of potassium. All right, let me kind of, made these a little bigger than I expected, but that's okay. All right, so now let's talk about it in the form of electrical properties. All right, and I'm going to try to think exactly how I want to do this, because I didn't do this as an exact copy of the way I wanted to, because I kind of opened this one a little too fast, brought these in a little too fast. But it didn't open up till there. All right, let's just do it like I got it. Okay. All right. I'm going to grab all these so it's the exact same. And then we'll just talk about the electrical properties here. Okay, so same kind of deal. Same exact thing. Although this time we're going to be talking about the electrical current. Okay, so what is happening here? So normally, let me actually draw one above this. Let me draw one above this too. Okay, and then I'm going to get rid of all this just to make it clear. Can redraw these in if I want. Okay. All right, and I got my channels. Get them bad boys. Put these up here. And slam this bad mamma jamma closed. Okay. All right, so normally, what is the membrane potential on the inside of the cell? It's going to be negative, right? Because there's less K on the inside, right? Because we're only pumping in 2 compared to sodium, which we're pumping out 3. So there's more positive outside. So it's positive on the outside, negative on the inside. So let's go ahead and just do that here. So it's positive on the outside. It's going to be positive. Positive. positive, positive. Okay? And then on the inside it's negative. Negative, negative, and negative. Okay? Beautiful. All right. Now what is happening is when the sodium rushes in like this, right? It's a bunch of sodium. It's not just one. It's a buttload. And so what has happened is we've flopped this. I'm going to go through. I'll just do it like this. It doesn't take that long. So now this is flopped. So the membrane potential is now positive there on the inside. On here it's on the outside still. Here it's on the outside. Here it's on the outside. Because now there's a bunch of sodium in addition to these potassiums. All right, and then that means over here now it's relatively negative. And okay, what that causes, this positive charge is actually what causes, on the inside is what causes this sodium channel to open. So we can write that. The positive charge on the inner membrane is what causes the sodium channel to open. Sesame seeds on a bun. All right. Okay, I think you can see where this is going. Okay, so then what happens here? Well, we had our sodiums rushing in. These sodiums were rushing in, if you remember. Right? And diffusing all along here. So what that means is we're going to have, again, positive all here, positive. And then we're going to have positive here as well. But it'll be positive up here, positive up here. Okay? Interesting. That means this will be negative, 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 negative. Okay, so it's actually these changes in membrane potential that cause these channels to open and close. It's the electrical charge across the membrane. All right? And when we've got both sides like this charge, this is actually what causes this to open, this sodium channel, after a while. It's kind of like on a delay. So when we've got positives here, this would be positive. It's now emptying. So this is still positive. Oops. So as that opens, it's going to start to drain away. This would be positive, and this would be positive. But now over here, it's actually flipped back to the way it should be. Back to good old resting, what we call resting membrane potential. All right. I know this stuff is very confusing. All right. And that's because this K here is rushing out of this area. Like so. Right. So now we're reestablishing, we're removing some of that positive charge to make this now relatively more negative on the inside and this relatively positive. Okay. Here we would have sodium rushing in, so it'd still be positive on both sides here until this closes, right? So we'd have positive rushing in, right, over in this direction to open up this channel. But also some of it, you know, would be coming over in this direction too, a little bit, which is why it would still be positive there. That makes sense. Okay? And then let's just seal the deal here with this last one. Now this is, over here it's the exact same. So it's positive, positive, negative, negative, which means, on the right color, I'm on positive. When that happens, when this reaches there on both sides, it's going to close this sodium channel. When the negative membrane potential is reestablished on both sides of the potassium channel, it'll close the potassium channel. This one would be negative on the outside. Just like when you're depressed or angry, you're negative on the outside. Oh, excuse me. This would also be negative on the outside. And this would be positive. Let me get rid of that. It's going to bother me. This would be positive. And we'll walk through this and make it make sense. Because I know you can get kind of lost in the weeds on this stuff. Okay. This is back to normal. Over here, we've got this opened up, which means we're having potassium rush out. So this would actually start to flip here. Right? And go back to normal just like it is. Boom. Boom. Right? To reestablish this. So this is normal resting membrane potential. And over here we've got our sodium mostly rushing in this direction to make it positive on the inside, but some would also, of course, be going in that direction, which is why it's still positive there. Okay? And it's this change in membrane potential, this distribution of negative and positive, that opens up potassium channels. Whereas it's just these positive, boop, boop, boop. You add positive to a sodium channel, and it's going to be open. All right? Okay, so let's walk through that again. Normal resting membrane potential. Normal resting membrane potential is positive on the outside, negative on the inside. Okay, beautiful. Okay. As sodium rushes in the cell, the inner cell membrane is now positive. Okay, so it flops it, it swaps it, it flips it, changes it, inverts it, makes it the opposite. Okay? Sodium channels are voltage, that's charge, voltage gated. They open or close their gates depending on the charge, and open when the inside of the cell is positive. All right, that make a sense? Pretty nifty indeed. Okay. Potassium, on the other hand, potassium channels are also voltage gated. And open when the membrane potential has been fully inverted or changed on both sides. Fully has been swapped after some time. Okay. Potassium channels open on a delay. That's what that results in, on a delay. And potassium rushing out of the cell re-establishes the resting membrane potential. Kind of a lot of goo. Okay, let's walk through it again. There's a lot of crap here to comprehend, but I want you guys to understand this because the very first time I saw this, I didn't understand it. And so I just really want to try to break this down as easy as possible. And it's not an easy thing to understand. It really isn't. So if you're struggling with this, if you're confused by this, if you're full of questions, that's okay. There's a lot going on here. But if you can remember, sodium rushes in, opens more sodiums, and then after a while, potassiums rush out to reestablish the charge across the membrane. If you can remember that and what the sodium-potassium pump is doing, you're golden, bro. Okay? So with the charge, you can just think of this as the charge. The charge flips when sodium's out, a bunch of sodium's on the inside. That opens up more sodium channels, which is then going to cause the charge to propagate down the neuron. So now it's positive here, which opens up more sodium channels, right? And now it's positive even further, which is going to open up this sodium channel. And as we get that kind of swap, eventually, of membrane potential, eventually what's going to happen is these potassium channels are going to open. Okay? And that's going to reestablish that membrane potential back to resting membrane potential. So it's a propagation of waves. It's an electrical signal being sent across the membrane and down the axon of the cell. All right? So all of this is happening within the axon of this cell here. All right, pretty cool. Lot there. Okay. All right, let's cap this off with a little discussion about how this all started, because we kind of skipped that and went right to the middle. Okay, how did this all start? Let's go over here. Let's go over to this little thing. And this little thing, let's see if I can pull a picture of this. Reminds me a lot of this. See if this will actually come into view here. Oh yeah, look at that. That's beautiful. Right, we've got, what I've got here is two neurons. So one neuron sends its axon down to another neuron's dendrites. And that's how neurons communicate with each other. So what we were talking about is how one neuron just sends signals down its body here. Okay? Like that. So now how does the signal jump from one neuron to the other? Or how does all of this nonsense I just talked about that confused the hell out of you, that you scribbled down in a notebook, and you really got to look back at this stuff to make sense of it. Okay? So feel free to rewatch this over and over again. In class, normally I'd review this part a bunch and we'd keep going over it and nonstop going over it. Okay, but what's going on here? Well, the signal somehow has to get between these cells. And this little part is a very special little part of our nervous system. And this is called a synapse. This is called a synapse. This is a synapse, okay? Where two neurons meet. Where two neurons send, we could say connect, but they don't really connect. Where two neurons meet. They don't really touch each other, but they meet here. Okay? They have a meeting. Very close, just like this. Oh no, I just ruined my life. Just ruined all of that. Dang it. It's okay. Okay, that's fine. I'll live, I'll survive. All right, so this over here, this would be an axon of the sending neuron, and this would be the dendrite of the receiving neuron, right? Let's zoom out and make sure I'm doing that right. Oh yeah, look, dendrites over here. Beautiful. That's a dendrite. This is a synapse. It's very much like this. Oh, look. Life is being injected into Adam. They're not actually touching, though. Pretty interesting stuff here, guys. All right? Okay. Same kind of deal here. All right, so how does this signal jump down? We talked about how it propagated. Let's talk about how it jumps down. So we can have two actually different kinds of signals. There's a lot of different types of signals, but we can have an inhibitory, which turns things off, or we can have an excitatory. Inhibitory, excitatory. All right, so let's talk about the excitatory first, because that makes more sense. A little easier to comprehend, I think. All right, so we got little things called vesicles. I don't know if you remember these. Good old vesicles. And these guys are full of, let's say, a little molecule, a little, let's say, protein. And that little protein is called a neurotransmitter. All right? Neurotransmitter. Not what I wanted. This would be a vesicle. You're going to want to draw this whole thing kind of big. Sorry if you've already started. Vesicle. Vesicle. And it's full of neurotransmitter. All right. An example of this would be an excitatory signal. Excitatory signal. And a version of that would be acetylcholine or acetylcholine. That's one example. Okay. What happens is as the action potential propagates down here, what it's going to do is cause these vesicles to start to fuse. So they're going to move, and start to fuse with this membrane here. So let's see if I can do this here in an elegant kind of way. This is going to look a little weird, but that's okay. Gonna do it like that. Okay. Looks a little fat there, that's okay. So this vesicle is now fused with the membrane and it's going to dump its contents, its neurotransmitter. All right, out into this extracellular space. And what that's going to do is it's going to bind to a little channel. That looks something like this. Okay. So this neurotransmitter in a vesicle fuses with the vesicle, and then it's going to bind to this little channel. Like so. And what that's going to cause the channel to do is actually open up like it is. So the neurotransmitter binds to channel and opens it up. Okay, crazy. Okay, that's pretty bananas. I don't know if you, do you remember this channel? It's blue, it's made out of 2 bars. Do you remember what that channel is? Oh, blue 2 bar sodium channel. Oh, maybe that's where this sodium came from originally. That magically just showed up and opened up this sodium channel. What do you think? What do you think, guys? You're telling me that your thinking is just the result of salt moving across channels in your cellular membranes? The fuck? The fuck, brothers? This is weird, dude. Even your best thinking is salty thinking? I don't know. So then sodium rushes in. I don't want it to be all fat like that. Right? Okay, and what's going to happen if sodium rushes in? It's going to change the membrane potential, which is normally what? It's normally negative. So now it's going to be positive membrane potential all in here, which is going to cause what to happen? Future sodium channels to open up. This positive membrane potential will open more sodium channels, causing the signal to propagate. Propagate. What? What? Okay. All right, that's an excitatory signal. This is an excitatory signal. All right, let's talk about an inhibitory signal. Well, it's still the same little story right here. We've got vesicles. They're full of stuff. That stuff is a neurotransmitter. Not too crazy so far. This one would be, for example, GABA is a inhibitory neurotransmitter. Boom, boom, and it's going to fuse with the membrane here. So we got our vesicle fusing with the membrane. So it goes like this. Fuses with it. I'm not doing that quite right. It doesn't matter. I don't care. I want to keep cruising. I know it's already gone along. But the membranes come together, they fuse. I don't know if you remember this. And let me go back. Look at that. Look at that crazy nonsense. Let's go all the way back to our cellular. Oh boy. Oh man, you remember this junk? Look at this. Oh, right. We had these little things called snares that are like bread ties that twist together to take this membrane and fuse it into this one. This is what's happening, right? Oh, and it's dumping out these proteins. So where are neurotransmitters synthesized? Not in the cytoplasm. They're synthesized in the rough ER. Oh, and they go to the Golgi. They get transported to the end on microtubules. Brother. Sometimes it's a burden. Like, just thinking about this, I'm doing that. That's the crazy part. Okay. All right. I'll stop wasting your time. I feel like I'm wasting your time, even though I'm teaching you things. Deal with it. Whatever. You're listening to this at double time. I had the thought of recording all of my lectures in normal speed, but then drawing them out to be double as long so that when you watch it at double speed, that's actually the maximum that you could watch it at is normal speed. Yeah. Who would want to learn at normal speed? Oh, it's kind of funny. Anyway, I'm not going to do that, but I could. All right, back to the matter at hand here. All right. We've then got a membrane channel here, which I believe I made these green. I believe these were green for potassium. Like so. Okay, so our neurotransmitter gonna bind something. These got to be up at the front. Yeah, they bind. It opens up that channel. Up to the top. Listen to me. Listen. You're not listening to me. Please listen. There we go. My life is it's a damn struggle sometimes. All right, so then these flow in. Okay. And when those flow in, I could have just drawn these as dang arrows. That's what I should have done. When these flow in, what happens? Oh boy, I'm struggling here. It all fell apart, right? What's going to happen here? These, no, sorry, I messed this up, sorry. This is actually a totally different channel, so I want to make these a totally different color. Might be. Okay. My thinking got all messed up there, I'm sorry. Let me rewind that. All right, let's go back to drawing just the channel and get rid of this guy. My brain got busted, I'm sorry. Okay. So we got our opening there. Now I need to draw the channel, and we're going to draw it in, let's draw it in pink, because this is a totally different ion actually here that ends up rushing in. Come on, you can do it. Beautiful. Beautiful. Okay, so this still binds nice and tidy like so. And this is actually for chloride. This is a chloride channel. So chloride rushes in. Don't want it like that. So chloride rushes in. And chloride, if you look at it, definitely negative. So all along here, this is all going to be negative now. And what membrane potential does the inside of the membrane inside of the cell usually have? It usually has negative. So neurotransmitter binds. Neurotransmitter binds. This would be inhibitory signal. And then this would be inhibitory. Neurotransmitter binds, opening up chloride channel. Which results in further establishing, basically like really makes the inside of the cell negative. Really establishing an inner negative membrane potential. It's pretty cool. That's pretty cool indeed. All right. All right. Couple of other, just like, let's do a little typey type. So this is how a synapse works, where two neurons meet. This is how the signal starts, and then it rushes down. The entire axon by opening and closing sodium and potassium channels, and then it gets down here, and the whole thing starts again. We got neurotransmitter release, sodium channels opening up, so on and so forth. Okay, let's just talk about some other, this will be the end, and this part's just some little typey type notes. Okay, let's just talk about some of our common neurotransmitters. All right. All right, just two, like some of the two kind of key ones that we think about, which would be dopamine and then also serotonin. So dopamine, dopamine. All right, let's put it up here a little bit further. So dopamine, and then we'll do serotonin. 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 Nope. Serotonin. Serotonin. Okay, not Sarah. Okay, and serotonin. All right, dopamine. This is motivation. Motivation. Not motivation, motivation. This is what gets you up in the morning. What keeps you going. So this is it's how you feel after you finish a huge project. You did something great and you want more. Okay? So this is why it's motivating, because like when you do it, you end up wanting more. When you get a lot of dopamine, you want more. And this is what drives us to continue to do things. You do it and you want more. And this is also really, really scary. Because like things like cocaine and alcohol and other drugs release dopamine, huge amount. It's crazy. If you actually go into an ice bath and then get out of it, you get a larger spike of dopamine than you do if you do cocaine. But cocaine dumps heck of dopamine in your brain, which is why you get addicted to it. It's like you just finished a huge project when you're on cocaine. Like you did something great, and then you want more of it. Okay, and then you actually get physiologically addicted to it. And alcohol is even scarier than cocaine, because alcohol only gives so it's like you've just done like a huge task, like you've crushed the day. You've done a to-do list, a whole page, and you like nailed it. Boom. I can go to bed tonight and hit my head on the pillow and like feel good about myself. Alcohol will only give you the checklist feeling of dopamine if your blood alcohol content is rising. That's why it's kind of scary. Because in order to keep getting that feeling, you got to keep drinking more and more so that your blood alcohol content keeps going up and up and up and up. Okay? It's a fake. Every time you take a drink, or you drink a beer or whatever, or have a whiskey, you're tricking your brain into thinking that it did something awesome, that it accomplished something. Kind of really takes away the value of alcohol when you look at it that way. It's a fake liquid, I just did a checklist feeling. Yeah, very interesting stuff. Okay, serotonin. Just a couple quick things on this one. This one is your satisfied neurotransmitter. Very pleasurable. You receive pleasure, satisfaction. You feel good. And you're done. You don't want any more. You don't want any more. It's like when you're full after you eat. Feeling of being full after you eat. I'm good. Okay? So this one's like contentment. Feeling satisfied. When you don't have this one, Low serotonin? Low serotonin. Oof. It's not a good place to be. I've been there quite a bit. Suicidal thoughts and aggression. It's a very, very, very bad place to be. Yeah, serotonin is huge for your feeling of like almost like I'm good enough. Yeah, I'm content in my brain chemistry. We could talk more about both of these forever and always, but this is, I think, damn near the longest one of these I've ever done, so I'm going to end it right now. I didn't even get to the tortilla thing. I'll put that in the next one. Okay. Dopamine, serotonin, acetylcholine, GABA, potassium rushes in, sodium rushes out, sodium potassium pump. We got a lot in that one. Proud of you guys. I'll see you in the next one.